Okay, welcome to the first video lecture for Chapter 7 on membrane structure and function. This chapter is a really important chapter because it deals primarily with cell membranes, or what we also call phospholipid bilayers. The reason why I say that this is such an important chapter is because the cell membrane represents the boundary between the living internal environment of the cell and the non-living external environment. As a result of that, we can't conceive of any way you can have a living cell, much less other more complex life, if you didn't have cell membranes. And so cell membranes then are just absolutely critical for life on Earth, at least as we understand it. The cell membrane or the plasma membrane is the boundary that separates the living cell from its surroundings, its external environment. But it doesn't entirely isolate the inside of the cell from the outside. Okay, things can move across that membrane, and as a result of that, we say that the membrane is selectively permeable. Selectively permeable. So permeable means that things can move back and forth across the membrane. The selective part means that not everything can, or at least the cell can control how fast and when things move across the membrane. So because membranes exhibit or show selective permeability, they sort of act like a filter. And because they therefore control or regulate what moves across, it's possible for a cell to maintain a very different internal chemistry than what you find outside the cell. There are some substances that pass across a membrane quite easily. Other substances, it is effectively an impermeable barrier, like a brick wall. Just depends on the substance, it depends on the situation, and we'll cover that as we go throughout this chapter. So let me show you sort of an animation here that will give you a feel for how membranes are put together and what they're like. So we talked in Chapter 5, I believe it was, about phospholipid bilayers. We were talking about phospholipids as a special kind of fat molecule, and I said that they're amphipathic, which means that one end of the molecule is polar, the other end, the hydrocarbon tails, are nonpolar. And as a result of that, being amphipathic, when you put them in water, they automatically form a bilayer structure. Well, in this video here, what you're seeing is the bilayer structure. This is water on the outside of the cell. Let's say this is water inside the cell. Here is a phospholipid. So you have the polar head, the nonpolar tails shown in green. Here you have the other layer, polar heads, nonpolar tails. We talked about how the tails back up to each other and create a fairly thick hydrophobic interior. Anything that's dissolved in water is therefore polar, right? We talked about that. It's hydrophilic and polar. Anything dissolved in water, or water itself also being a polar molecule, is going to have a difficult time getting through that fairly thick hydrophobic region. So what we have here in the middle is a protein. It's called a channel protein. We'll see more about those in this chapter. But this protein here is going to allow for water molecules to move through much more quickly than if they try to simply diffuse or squeeze between these phospholipids. So let me play this. This will also give you a nice feel for sort of the liquid or the fluid nature of membranes. So you can see everything jostling around, the water molecules, all that, but you can see the water molecules also flowing in or out through that protein there in the middle, okay? That's basically the structure of a phospholipid bilayer. Okay, so again, those phospholipids, we'll say it again, are amphipathic molecules, meaning they have a polar region and a nonpolar region. They have a hydrophobic region and a hydrophilic region, okay? As a result of that one thing alone about phospholipids, they form that bilayer that we talked about. So let's take a look. So here you see a phospholipid bilayer, figure from your textbook, okay? We see a phospholipid here, for example, one there. Again, you see how the tails are backed up. But if you zoom in, if you could see this at the level of the individual molecules, but you can't, you might see something like this. This is the hydrophilic or polar head. Remember, there's our portion of the molecule that the phospholipids actually attach to, okay? And we can see that there's no water down here at all because that's a fairly thick hydrophobic region. Up here you have water molecules, and it's this portion of the molecule that gives it that, or that has that charge, which is something therefore that water can attract to. Same thing down here. Remember that glycerol portion of the fat molecule is nonpolar, just like the tails are, so water's not attracting there. In a typical lipid, that's all you'd have. You wouldn't have these extra attachments here, the phosphate and the choline, which give it those charges. But in phospholipids, you do have those, and that's why the water molecules attract. Okay. But again, because the hydrophobic tails there in the middle want to repel away from water, they sort of swing inwards and back up to each other, forming this relatively thick hydrophobic layer. It's important to realize that anything dissolved in water outside or inside the cell is hydrophilic, and therefore will not be able to very easily get across that hydrophobic layer. It's the same effect as oil and water. They don't mix because one's hydrophobic and one is hydrophilic. Okay? So this hydrophobic interior is a large part actually of why cell membranes are selectively permeable. There are other factors involved, but that's a big one. So what I showed you in that previous little animation was a good representation of what we call the fluid mosaic model of membrane structure. Okay, the fluid mosaic model. This basically states that a membrane is a fluid structure with a mosaic of various proteins embedded in it. It's fluid in the sense that the phospholipids that make up that membrane are not bonded to each other. They're just sitting there sort of loose, held in place by hydrophilic interactions with water. All right. There are proteins embedded in that membrane, and they represent the mosaic part of it. Okay. Now, these proteins are not randomly distributed in the membrane, and it's also true that different cell types will have different assemblages of various proteins in their membranes. The proteins can tell you a lot actually about the various functions of the cell. So different cell types will have different groupings or different types of proteins embedded in their membrane. Okay. The fluid mosaic model is a conceptual model which tells us that the membrane is relatively fluid. Okay, that's the phospholipids moving around relative to each other, but it also has a series of proteins or a bunch of proteins embedded as the mosaic part in that fluid structure. So let's take a look at a fairly nice illustration from your textbook about this. So here you can see we have our phospholipid bilayer, right? And we have, like we talked about in the last chapter, the ECM on the outside. So this is an animal cell. We have the cytoskeleton with the cortex of microfilaments on the inside, again, all tied together. We have integral proteins that span the distance from one side of the membrane to the next. They're embedded in that hydrophobic interior. And in some cases, fibronectins attach to those for attachment to collagen or attachment to the ECM or the cytoskeleton on the inside. But you see we have a large variety of different kinds of proteins embedded in that membrane, and that's essentially a representation of the fluid mosaic model, our current conceptual understanding of how proteins, or how membranes rather, are put together. Okay, so let's go back really quick to that fluid idea of the membrane. Let's imagine that you're washing dishes in a sink and you have a layer of soap bubbles, suds, a suds layer on top of the water. You could put your finger in that suds layer down into the water, move your finger around, and the suds would move out of your finger's way and sort of reform behind it. We would say that soapy suds layer there is sort of fluid. Now imagine that the soap bubbles, are not soap bubbles, but they're phospholipids of a cell membrane. If you could put your finger into that cell membrane in between the phospholipids and move it around, they would move out of your finger's way and sort of reform behind it. It's a fluid structure, all right? But again, embedded with these proteins that give it the mosaic aspect. So these lipids then can sort of drift laterally back and forth, left and right, in a very fluid sort of way, okay? And sometimes they may even flip-flop from one side of the membrane to the other, but that's not as common apparently as moving around in a lateral sort of way. Now, what about the effect of temperature on how fluid the membrane is? Well, this is an important consideration because membranes have to be fluid in order to work properly. In other words, in order to be selectively permeable and to actually allow things to move across when things need to go across, the membrane has to be fluid. but not too fluid. It has to be just right, in other words. So temperature can certainly affect this. As temperatures cool, membranes tend to switch from a more fluid state to a less fluid or possibly even a solid state. Think about bacon grease, right? Let's say you're cooking bacon, and when you're done cooking the bacon, the grease is hot, it's liquid. But if you leave it sit there for a while on the griddle, it will eventually solidify and turn into that white sort of pasty material at lower temperature. That's a good example of how temperature can affect the fluidity of cell membranes, because, like the bacon grease, cell membranes are made mostly of fat, phospholipids. To what extent the membrane is composed of unsaturated versus saturated phospholipids can also play a role in determining how fluid the membrane is, in addition to temperature. So membranes that are rich in unsaturated fatty acids are more fluid than those that have a higher proportion of saturated fatty acids. And again, membranes need to be fluid to work properly. So one way that our cell membranes kind of fight against this tendency to become more or less fluid as temperature changes or whatever, and also with regard to how much saturated fat is in the membrane, is through cholesterol. You see there's cholesterol molecules here embedded in our phospholipid bilayer. And cholesterol plays two important roles here. At high temperature, when the membrane would tend to become too fluid and could possibly dissolve or fly apart, which would be bad, that would mean cell death, the cholesterol constrains the phospholipid movement and prevents them from flying apart from one another. So it keeps the membrane intact at higher temperatures. At lower temperatures, cholesterol prevents tight packing of phospholipids, which would tend to make the membrane more solid. And so by preventing that tight packing, it keeps the membrane fluid at lower temperatures. So the effect of cholesterol here then is to broaden the range of temperatures across which the membrane can remain fluid, so it remains fluid at higher and lower temperatures than it would otherwise with cholesterol present, and the cholesterol therefore helps the membrane to be selectively permeable under adverse conditions of temperature. Okay. So let's take a look at the effect then of cholesterol, but also how much of the membrane is saturated versus unsaturated phospholipid. So here we have a really fluid membrane. Okay, you have a higher proportion of unsaturated phospholipids. Remember, they have an unsaturated fatty acid chain. The double bond there, which is why we call it unsaturated, gives it that characteristic bend or kink. Well, if you have a higher proportion of phospholipids that are unsaturated, they will push the next phospholipid further away because they have that bend or kink. And so you have a membrane that is less densely packed with phospholipids than here, where you have all saturated fats where the tails are straight. You see how those will pack in more tightly. This is a more viscous or more solid sort of membrane. Viscosity refers to how easily a fluid flows. So compare honey and water, right? Water flows very readily compared to honey. Honey is more viscous, water is less viscous. This is a more viscous membrane. That's a less viscous or more fluid membrane. Then here you have cholesterol sitting between that membrane that has a higher proportion of saturated fats, and so it helps to keep that membrane more fluid when because of the phospholipids, the saturated fats that it has, it would tend not to be fluid. Okay, so we said that a membrane then is sort of a collage of different proteins that are often grouped together, embedded in that fluid matrix of the lipid bilayer. Okay, sometimes they're attached to one side or the other of the membrane, sometimes they go all the way through. In the case where they go all the way through, we call those proteins transmembrane proteins. Okay, transmembrane proteins, they go all the way across the membrane. And as I said, proteins determine most of the membrane's specific functions, and therefore you can understand a lot about what a cell does by understanding what kind of proteins are found in its membrane. So, first of all, we have proteins that are just attached to one side or the other, as I said. Those are called peripheral proteins. Peripheral means on the side or on the edge. These are proteins that are just bound to one surface or the other of the membrane. Now, those proteins that are embedded in the membrane and go all the way through that hydrophobic interior They are also called integral proteins, or as I said, transmembrane proteins. Either term is fine, but these are proteins that penetrate that hydrophobic core and sit there in the middle of that membrane, with part of them projecting out on either side. So these transmembrane or integral proteins are very important, and it actually turns out they're not bonded to the phospholipids around them. Okay, they're not bonded to the phospholipids around them. They are locked in place by hydrophobic, hydrophilic interactions. Remember what proteins are made of. What are the monomer building blocks of proteins? Amino acids, right? We talked about that in chapter 5. And we said that some amino acids are polar, some are nonpolar. So some are hydrophilic, they attract to water. Some are hydrophobic, they don't. So when you have a polypeptide, many amino acids will be nonpolar, or regions will be polar, and some will be nonpolar. And it's those interactions that will lock that protein that polypeptide folds up into that spot in the middle of that membrane so it doesn't come out. The hydrophobic regions of an integral protein will usually consist of one or more stretches of nonpolar amino acids, and these are often coiled into alpha helices. We talked about that as one of two types of secondary structure of proteins back in chapter 5. So, take a look at this example. So here you have your phospholipid bilayer, and you have a transmembrane or integral protein. Okay, here you can see you have alpha helices, several of them. This is a protein that's going to provide sort of a channel for something to move through, like a tunnel. Okay, so these alpha helices are kind of arranged in a cylinder. You could look straight down on top of that, like looking into a pipe, for example, with the center being hollow. A tunnel, in other words. All right, let's imagine that these amino acids here are hydrophilic. They attract to the water outside the membrane. Let's say these amino acids are hydrophilic. They attract to the water inside the membrane. These amino acids here in the middle that are interacting with the hydrophobic tails of the phospholipids would be hydrophobic. So if you could sort of grab onto the bottom of that protein with your hand and try and pull it down, so try and pull it down out of that membrane, what would happen is a couple of different things. First of all, these hydrophobic amino acids would get pulled down and start to interact with water, and they would repel each other. Also, these hydrophilic amino acids would get pulled into the interior of the membrane and start to interact with hydrophobic tails of phospholipids, and they would start to repel each other. So if you're trying to pull this down, you would start to feel resistance. If you let it go, it would spring right back up into its original spot and just stay there, hang there in the middle of that membrane. It's these hydrophobic, hydrophilic interactions that are locking it in place. All right, that's a good example of a transmembrane or integral protein. And we'll see lots of examples of those. We are actually going to be talking about the various categories of membrane proteins in terms of their function next. Okay, so when you look at all the different kinds of proteins you find embedded in membranes, okay, or even those that are just attached to one side or the other, they can basically be broken down into 6 different categories. We have what are called transport proteins. Transport, those are just going to allow hydrophilic substances, let's say, that can't get through the hydrophobic interior of the membrane, a route of safe passage from outside the cell or inside, whichever way it's going. We have proteins that are involved in enzymatic activity. So proteins that act as enzymes that help to run chemical reactions. We have proteins that are important for what's called signal transduction. This is nothing more than passing messages from inside the cell to out, or vice versa. Okay? Usually outside to in, actually. We have proteins that are involved in cell-to-cell recognition, allowing cells to recognize one another. We have proteins that are involved in intercellular joining, helping to join cells together through their extracellular matrices, okay? And as we've already seen, we have proteins that allow for attachment to the cytoskeleton and the ECM. We talked about fibronectin, for example, in the last chapter, okay? So most membrane proteins can be lumped into one of these 6 categories, and we're going to talk about these in a little more detail here. Okay, transport proteins. And in fact, transport proteins are what we're going to spend a large part of the rest of this chapter talking about because they're so important in helping to determine how things move into and out of a cell. So transport proteins, we have proteins that act as simple tunnels, allowing things to diffuse from high concentration to low through the tunnel into the inside of the cell, let's say. Or we have proteins that act as pumps, using the energy of an ATP molecule to pump something from low concentration back to high, which is the reverse of the natural flow of things, as we'll see. So transport's very important. Enzymatic activity, taking an initial reactant, converting it to some intermediate molecule, and then finally to some final product. Usually enzymes are sort of embedded, in a row like you see here in a membrane, and each one will run some part of that overall multi-part chemical reaction. We'll see good examples of that in chapters 9 and 10 with respiration and photosynthesis. Signal transduction. Here you have a protein that acts as a receptor in a cell membrane. Here you have a signaling molecule. Maybe it's a hormone of some sort. You have a gland in the body that releases a hormone into the bloodstream. That hormone has a target tissue somewhere in the body. In that target tissue, the cells have these receptor proteins. That hormone binds, and this does not transport the hormone into the cell. The hormone just binds to one side of it, outside the cell. That causes a change in shape of the protein, which causes a chemical reaction inside the cell. It's called a cascade. It's like a chain reaction. That cascade or chain chemical reaction inside the cell causes the cell to do whatever the message is telling it to do. Cell-to-cell recognition. Here we have a group of proteins I haven't mentioned before called glycoproteins. They have a small sugar on one side, hence glycoprotein, sugar protein. And the sugar here will bind to a receptor protein on another cell. So for example, with our immune system, we have macrophages, for example, white blood cells, for example, that will bind with your regular cells in your body because your cells have the correct glycoprotein on them. If a bacterium gets into your body and that protein on that macrophage or white blood cell can't bind to it, that's a signal to the macrophage that it's something foreign and needs to be destroyed. Okay. Here we have intracellular joining. In some cases, you'll have cell membranes of two adjacent cells sort of spot welded together by a bond between these two proteins that are in their adjacent cell membranes. Okay. In many cases, that helps to sort of zip cells together in watertight connections, things like that. All right, we have various different types of connections like that. And then as we've already seen, maybe you have a fibronectin protein here attached to collagen of the ECM. That's connected to a transmembrane protein, and then that protein is connected to the cytoskeleton on the inside. So these proteins tying the ECM to the cytoskeleton through the cell membrane. Okay, so let's talk about, go back to this idea of selective permeability of the cell membrane. It's very important because it helps to regulate and control movement across the membrane, as we've said, allowing the membrane to act as a filter. So a cell has to exchange materials with its surroundings, and this is largely controlled by the cell membrane, the plasma membrane. Plasma membranes, being selectively permeable, are therefore regulating the cell's molecular traffic. Okay? And so this is where this idea of hydrophobic and hydrophilic molecules is very important. Remember, a hydrophobic or nonpolar molecule is not going to be attracted to water, but it can be attracted to other fats, like dissolves like. If you take corn oil and olive oil and mix them together, they will mix because they're both nonpolar and they'll mix with each other. There's no repulsive effect there. Now, hydrophilic molecules, likewise, when in coming into contact with each other, they'll attract to each other as well. Okay. Remember, like dissolves like. Hydrophilic dissolves hydrophilic, hydrophobic dissolves hydrophobic. But hydrophilic and hydrophobic won't mix, they repel. Okay? So, hydrophobic molecules like hydrocarbons can dissolve in the lipid bilayer and pass through the membrane pretty rapidly. Fat molecules can go through that membrane pretty easily because that interior hydrophobic region presents really no barrier to them, like dissolves like. Hydrophilic Polar molecules, on the other hand, like ions, large sugars, that kind of thing, do not cross the membrane very easily. So they might well need a transport protein to allow them to move through. So transport proteins are very important for allowing hydrophilic polar substances to move across the membrane, inside to out or outside to in. So we have some transport proteins that are very simple. They're just called channel proteins because, again, they behave as nothing more than a tunnel, right? As long as a solute, a chemical dissolved in water, can fit through the opening of the protein, it can go right on through. There's a special category of these channel proteins, though, called aquaporins. Okay, aquaporins. And these transport water. They specialize in transporting water. Aquaporin, water pore. Okay, so these are like floodgates in a cell. There are times when a cell needs to get rid of a lot of water fast or take on a lot of water very fast. And so when that happens, the cell will open these aquaporins and allow things to move through very, very rapidly, water to move through rapidly. Okay, now there are other transport proteins that are called carrier proteins. Carrier proteins will bind to molecules and change shape to sort of escort that molecule across the membrane. Okay, they're quite selective. They're very selective. A typical carrier protein may only move one kind of molecule across that membrane. Usually the molecule and the carrier protein will fit together like puzzle pieces, or at least recognize each other based on their shape. Okay, and this is how a carrier protein can only move one or maybe just a few different types of molecules and not let everything move across. And we'll see more examples of carrier proteins here in a little bit. All right. Okay, but now we need to shift gears a little bit and sort of go back to bigger ideas about how things move across membranes. The simplest way for something to move across a membrane is through what's called diffusion. Okay, diffusion. Diffusion is also referred to as passive transport. Okay, diffusion or passive transport. Diffusion is a tendency for molecules to spread out evenly into the available space. Remember, we talked in an earlier chapter about how all molecules are moving, and the higher the temperature, the faster they move, the lower temperature, the slower they move. So molecules are jostling around, vibrating around, bouncing into each other, and then spreading away. When they impact with each other, they go off in opposite directions. Think about pool balls on a billiard table, right? So, because molecules have heat and they're moving around, if you have them highly concentrated in an area, they will bump into each other and begin to spread away from that common center. That's diffusion, okay? So each molecule is moving randomly, but diffusion of a population of molecules may be directional, typically away from an area of high concentration, okay? Now, that's an important rule, okay, in nature. Things move from high concentration to low, okay? Things move from high concentration to low until there's no longer a difference in concentration. So if we have a difference in concentration of a solute, let's say on one side of a membrane versus another, that is what we call a concentration gradient. Concentration gradient. Gradient just means difference in something. Concentration gradient is difference in concentration, okay? Nature doesn't like concentration gradients. Nature will try to eliminate concentration gradients. So if you have a high concentration of solute in one area compared to everywhere else around it, those molecules of solute will diffuse from high concentration to low. They'll spread out. This is why a sugar cube dissolves in water without you having to stir it. It's why dye will mix evenly with water after you drop it in there. Initially it's there highly concentrated as that drop, but it spreads on its own because the molecules of water are vibrating, as are the molecules of dye, and just spreads. Important point about diffusion is that it requires no energy to happen. The energy that's already there is the heat in the environment causing the molecules to vibrate and smack into each other and spread away from a common center, okay? No energy is needed for this to happen. So let's take a look. So let's say we have two volumes of water, one on the left, one on the right, separated by a permeable membrane, one with openings as you see here. If you drop a yellow food dye, food coloring into one side, those molecules vibrating around will eventually find their way through the openings and start to diffuse from high concentration to low. We have a gradient, high concentration, low. That's a concentration gradient. So these molecules will diffuse this way. Occasionally one will go back the other way, but it's outnumbered by the ones going this way, down the gradient, until eventually you have the same concentration of ions or solutes or whatever on both sides. That is called equilibrium. In particular, dynamic equilibrium, because the molecules, even though there's no longer a gradient, that's equilibrium, they're still moving. That's dynamic. If one happens to go back to the left, it's counterbalanced by one that goes to the right. You always have the same concentration once that concentration gradient is eliminated. And you can have two molecules moving in opposite directions down their respective gradients. So you have, again, yellow food coloring on the left-hand side, but now you put purple on the right. Now you have two gradients, one for yellow to the right, one for purple to the left. They both will diffuse across the membrane down their gradients, yellow this way, purple that way, until you get the same concentration of both on both sides. So we have two gradients that have gone away there, and we have two different situations of dynamic equilibrium for both molecules. It's important to recognize that this dynamic equilibrium will never, on its own, go back to a gradient, okay, without the application of energy. If no energy is applied, this will stay this way forever. It won't spontaneously go back to this. That'd be akin to a building itself without a construction crew. That doesn't happen. But as we're going to see in a little bit, we can have cells that create concentration gradients, but they have to use energy to do it. All right, so here is an animation on diffusion. We'll play this.
Diffusion is the tendency for particles of any kind to spread out from where they are more concentrated to where they are less concentrated. This process can also be described as molecules moving down their concentration gradient. Diffusion across a biological membrane is called passive transport, since the cell expends no energy to move the molecules. Oxygen and carbon dioxide molecules move into and out of cells by passive transport.
Okay, so again, importantly, it's referred to as passive transport. Diffusion is referred to as passive transport. The passive part implies that no energy is needed to make this happen. Okay, you don't have to add any more energy. It's already taking place because of the energy present in the form of heat that's in the system. Okay, and it causes gradients to disappear, as you see there. Now, let's look at another animation on membrane selectivity. Why it is that membranes are selectively permeable.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions, such as sodium ions and hydrogen ions, cannot pass through the membrane unaided.
Okay, so bottom line here then is that substances diffuse down their concentration gradients from high concentration to low. If you happen to have a substance in high concentration outside the cell and low inside, it will want to diffuse from high to low to the inside of the cell. Now, the question is, can it get across that membrane? If it's a hydrophilic polar substance, it can't get through the membrane directly, usually. It'll need a channel protein or a transport protein to move through. If it's a fat molecule, or if it's hydrophobic and nonpolar, it can diffuse across that pretty easily down its gradient. Okay, the next topic is going to be osmosis, and I think that this represents a really good stopping point before we get into that. So we're going to stop here. The second part of this video lecture for Chapter 7 we'll pick up with